My name is Li Zhou. I'm a professor of pathology at MGH Cancer Center. And I'm a basic scientist, not a clinician. So the research of my lab is actually focused on understanding how cells respond to DNA damage and how some of the DNA repair proteins, including BRCA1, BRCA2, and this kinase called ATR, how they function in DNA repair. So what we have learned from our study is that when BRCA1, BRCA2 mutated cancer cells, when they become resistant to PARP inhibitor, they actually somehow rewired their DNA repair pathway to bypass these important proteins, and so that even in the absence of BRCA1, BRCA2, they can carry out homologous recombination, which is important for repair of DNA breaks. And they can also protect their stalled DNA replication forks, and therefore they are no longer sensitive to PARP inhibitor. So we're learning about all this rewired DNA repair pathways. And the exciting finding we made was that even when cancer cells rewire their DNA repair pathways to bypass BRCA1, BRCA2, they still need this kinase called ATR to repair. And therefore, when we use specific inhibitors to eliminate ATR in those cancer cells, now all of a sudden they become sensitive to PARP inhibitor again. So we think that even though this is still kind of a finding from basic research, this combination of ATR inhibitor and PARP inhibitor may be particularly effective in overcoming PARP inhibitor resistance in patients. So this is our main finding and the hope for future clinical trials. Yes.